Hello and welcome to part 2 of the overview lecture on chapter 6. We're going to now take a look at more detailed overview of immunity. As we've done before, we'll keep putting this diagram up and I'll indicate in red where we're going next. So we saw in the part 1 that most of the cells came from the myeloid line. Let's take a look at some more detail about those cells. As I mentioned in part 1, the mast cells are one of the most important cells. The mast cells are some of the cells that had those inclusions. They have bubbles inside, referred to as granules. Those granules contain histamine, and it's histamine that causes a lot of the reactions that we think of, or a lot of the symptoms associated with inflammation. So the vascular smooth muscle cells respond to histamine by relaxing. And so that's why you get vasodilation of the blood vessels in the area. Additionally, the epithelial cells, the endothelial cells that line those capillaries, they kind of pull away from each other, and that allows for more permeability. So liquid that's in the capillaries can come out into the tissues, and we get swelling, which we associate with inflammation. So vasodilation allows more blood to flow through the area. So there's redness, there's more heat in the area. These are all cardinal signs of inflammation. And it's all in reaction to histamine. Mast cells also can release, so once they're done releasing the histamine that they had stored up, they can also produce leukotrienes and prostaglandins that have some of the same effects as histamine. So there's vasodilation, there's increased capillary permeability. Oh, another one that I did not mention with histamine, but is also true for histamine, is the smooth muscle that's in the vasculature that relaxes, but the smooth muscle that's in the respiratory system that contracts. And so we actually get bronchoconstriction at the same time that we're getting vasodilation. So it's the same signal molecules, but the receptors are causing different things to happen in the different kinds of cells. And for those of you who are aware of medications that are antihistamine, they block this signal so that we don't have all the inflammation. And prostaglandins. The additional trick that prostaglandins have is that they stimulate the nerve endings to create pain. And it's things that block the production of prostaglandin that help us avoid things like NSAIDs. Block the production of the prostaglandins. Amazing. All right, let's continue with some of these innate immune cells, neutrophils. We know them and love them. They're the most abundant white blood cell. They specialize on a bacterium, and specialize, I mean, they respond to the whole group of bacteria. When we see a complete blood count and we see neutrophil numbers are elevated, that tells us that the patient is having an acute infection. It's early on in the infectious process. So we get an increase in the white blood cell count, leukocytosis. These are highly mobile cells. They actually can leave the blood vessel. They're the coolest thing. They actually slip out between the endothelial cells and can kind of move like amoebas and go after bacteria. It's fantastic. Then monocytes, these are also white blood cells. Their name changes because they change themselves when they come out into the tissues. They can mature into macrophages, big eaters. These are huge and phagocytic cells. Neutrophils can only consume so much. And macrophages can take in a lot more. Macrophages are much longer lived than neutrophils are. Neutrophils will last a day, maybe, and macrophages can last longer than that. Let's see, they migrate into the tissues. These are the professional phagocytes. In the case of chronic inflammation, you're actually going to see an increase in the monocyte number. And so that tells you that we're further along in the infection, or we're further along in the inflammatory process. Both macrophages and dendritic cells function as antigen presenting cells, which we'll talk about again later. They present things to the helper T cells. And then in passing, I'm going to mention eosinophils. So eosinophils, their job is to respond to multicellular parasites, things like parasitic worms. They don't have a lot to do in this country because we don't end up with a lot of parasitic worm infections around here. And we have been found that they're the leukocyte that's most associated with allergic reactions. And that's actually one hypothesis for why we have so many allergies in this country, is that we didn't get exposed to worms early on in our lives. And so the eosinophils, they're looking for a job, and so they overreact to stuff. That's just one hypothesis. Some information about these cells can be found on these pages. And then there were some innate immune cells, excuse me, that came from the lymphoid line, and I'll mention those briefly. These are the innate lymphoid cells, the ILCs. We're not going to use that acronym. You will see NK, natural killer. These were the first lymphocytes that were shown to act like innate immune cells. They can go after virus infected cells. So our own cells that have been infected with a virus, they have to be tagged by some other cell first. And then the natural killer cells can go after them. And they can go after transformed self cells. In other words, cancerous cells. And there are some others that have been identified. For the purposes of our class, we do not have to go into those details. Information about these is on those pages. And now we're going to shift over to, we're still with cells, but now let's take a look at these B cells and T cells, the lymphocytes. The lymphocytes that act in a very precise way. These are the so-called B cells that mature when they are stimulated. All right, so let's see, I have to back up a little bit. B cells and T cells have structures on them that match very specifically to the shape of something that has come in from outside or something that's inside our bodies. Each B cell and each T cell has its own thing that it responds to, its own shape that it's sensitive to. And if you're very interested in the details about how you get so much variety, we can go into it. Our textbook goes into enormous detail about it. We are not going to go into that detail unless you're very interested and you want to ask me some questions about that. Otherwise, let's just say B cells and T cells, highly specialized for a particular shape of something, and we're not going to get into how. It's the B cells that if the one that's sensitive to a particular thing gets stimulated by that particular thing, that cell matures into a plasma cell and then goes on and produces what are known as antibodies. The T cells, they come in a whole lot of different kinds, and our textbook does go into that in some detail. I'm going to expect you to know 3, and 2 of them are related to each other. So cytotoxic T cells, these are the ones that can actually go around and kill cells. Either if they're going around, well, there's some variation in how they do it, but some cytotoxic T cells directly kill the cell, and other cytotoxic T cells stimulate a self cell, one of our own cells, to undergo apoptosis. That would happen, for example, in a cell that's precancerous, starting to look like things aren't going right in that cell. The cytotoxic T cell basically presses the button and says, yeah, go ahead and self-destruct, because you're not going to do good things for us. And then I want you to know that there are so-called helper T cells. The helper T cells have been shown now. There's 5 different categories. Our textbook describes them all. I don't need you to know all that detail except for this. There is one class of helper T cell called a regulatory T cell, or Treg. It turns out, remember we said sometimes the immune system can create problems and go after cells that it's not supposed to go after. Well, the regulatory T cells are the ones that are supposed to nip that in the bud, like get rid of the cells that are reacting, and basically like turn them off and tell them, do not keep doing what you're doing. It is believed that it's an accident, a mistake, something that has gone wrong with the regulatory T cells that is to blame for some of the autoimmune conditions that we see. That in fact, had the regulatory helper T cell been functioning properly, that the autoimmune reaction would have been cut off and eliminated. Okay, we'll get back to that. All right, I did mention that there is cooperation between the cells of inflammation and the cells of adaptive immunity. And so one of the ways that they cooperate is the cells of innate immunity. Let's see. Remember macrophages and neutrophils, they're phagocytes, they eat other things. Well, one of the ways that they know that something needs to be eaten is because they find it covered in antibodies. The antibodies are sticking all over this thing, and we call that opsonization or opsonizing, and that's how the macrophages and neutrophils know that that's something that they have to go after. There are other chemical signals that are released by the lymphocytes that stimulates the inflammatory response. And then the other way they interact is the lymphocytes need to have antigens presented to them. And it's those antigen presenting cells, macrophages and dendritic cells, that say, here, go after this. If your piece is sensitive to this shape, go after it. And I think I showed that on the next slide. Okay, yeah, so here we're going to see how the cells of innate immunity and adaptive immunity can interact with each other. And this diagram I took from your textbook. Let's see, over here on the right, we have the cells of adaptive immunity. There's some B cells and some T cells. I'll get back to the CD4 and the CD8. CD4, these are helper T cells, and CD8, these are cytotoxic T cells, and I'll tell you how to remember that. And so we mentioned the antibodies. The antibodies will coat a bacterium, for example, and the macrophages and the neutrophils can respond to that. There are chemical signals coming from the lymphocytes that triggers the natural killer cells to respond and destroy things. And then here's the antigen presentation. The dendritic cells and the macrophages can both do this. They present antigens to the lymphocytes so that they'll go after them. So yeah, lots of interaction. All right, so we're here. Now we have finished with the cells for now, and we're going to move on to the inflammatory chemicals. I mentioned some of them in passing in part 1, but some of the mediators of inflammation include histamine, these from mast cell degranulation. For those of you who know detail and want detail, basophils also release histamine, but we're not gonna, that's a detail, don't need to know it. Prostaglandins and leukotrienes are also released by mast cells, but later, after they've already degranulated, they've already released their histamine, they start producing prostaglandins and leukotrienes also. And then there's some other chemicals that are produced by other cells that they can respond to. Okay, so what happens in response to histamine, prostaglandin, leukotrienes? We already said the vascular smooth muscle relaxes, so the blood vessels dilate. The capillaries get leaky. So fluid goes out, but more white blood cells can also come out more easily. We get more blood flow. The plasma proteins of the complement system can leak out into the tissues where the infection is, and release white blood cells to the injured area. And this is when the cardinal signs of acute inflammation develop: the heat, the redness, the swelling, the pain. And then ultimately, all that increased blood flow helps with tissue repair and wound healing. This diagram is quite simple. It's not in your textbook. I included it in the PowerPoint so you can take a look at it more closely. It's from the Understanding Pathophys book by Heather and McCants and Brashers. Very briefly, you can get inflammation to be triggered by just tissue injury or tissue injury and pathogenic invasion. You get the mast cells to degranulate, they release their histamine, and look at all the effects. This is not a great diagram of this, but what this is supposed to be showing is arteriolar vasodilation, so the relaxation of the muscle, so you get more blood flowing through the area. This also allows the cells that are lining these capillaries to pull away from each other a little bit and let fluid come out, the fluid that contains the complement proteins, as well as some of the white blood cells can squeeze out here too. Oh, here they are, showing cellular infiltration by the neutrophils. Yeah, this isn't by accident. These neutrophils are changing their shape intentionally and moving. It's just fantastic. Blood clot formation, going back to the protein systems. We mentioned complement, but the clotting system also gets activated to close up the holes. And then finally, we mentioned the stimulation of the nerve endings by the prostaglandins, creating pain in the area. And pain's not fantastic, except that it is a great early alert system. It's notification to the nervous system, something is wrong. If you have your hand on a stove, take it off right now, or move away from the pointy stick, or whatever. There's the thorn in your claw, or in your claw, good Lord. There's there's something in your foot, take it out. Okay. All right. And so we mentioned the plasma protein systems. We're going to focus on complement system. Our textbook has its own description. On this page, there's a box that goes through complement in some detail. This is quite good at killing bacteria. So it's similar to the coagulation cascade that you've probably learned in some detail, in this sense that the proteins are already present in the circulatory system, but they're in an inactive form, and they can get activated in one of three ways that we're not going to go into. What I want you to know is we keep discovering new ways. So there was the classical pathway, there was the pathway period, and then somebody discovered this other pathway, and so that got called the alternative pathway. Then somebody else years later discovered the lectin pathway. I mean, I don't doubt there's going to be more pathways that will be discovered, different ways that this gets triggered. Once it is triggered, there is this cascade of proteins activating the next protein, which activates the next protein, and each of those has an impact. let's see, so some of the things that happen, it triggers more release of histamine from mast cells. It causes more vasodilation. It causes more white blood cells to leave the bloodstream and go out into the tissue where there's an infection. This is like the grand finale of the complement cascade. There is what's known as a membrane attack complex, or MAC or MAC. And this, oh, you know what, before I get to that, I'm sorry, I just saw up here, coating of bacteria with the complement proteins, just like the antibodies sticking to bacteria, the complement proteins can stick to the bacteria and also act as opsonins for neutrophils and macrophages to go and consume those things, promote phagocytosis. And then finally, here's the grand finale, the formation of this membrane attack complex. And why I say grand finale is because there's a whole bunch of things that have to happen, and then they have to happen quickly at the end. A bunch of these proteins actually get embedded into through the cell wall and cell membrane of this bacterium, and they make a hole so that fluids go rushing into the cell and the cell lyses. It's fantastic. All right, so that's how complement works. All right, now we're going to turn our attention. We finished here with plasma proteins, and now we're going to look over at adaptive immunity. You'll remember the cells here are from the lymphoid line, the B cells and the T cells. So the immune system, adaptive immune system, anatomy, the primary tissues that produce the cells, the bone marrow and the thymus gland. The other tissues are secondary, meaning they don't produce the cells, but the cells go do something there. So the lymph nodes, tonsils, the spleen, the Peyer's patches. This one used to be, I used to find this quite confusing as a student, but let's see if you can think about it this way. You have the epithelial tissues on your skin, and you have the epithelial tissues of the mucous membranes. And those are the tissues that are most exposed to the outside world, right? I mean, mostly we think about our skin, but you know, lining of the mouth and the eyes and the lining of the nose, those are exposed to smoke, or you know, whatever might be coming in, infectious agents, breathing in the lungs, you know, the mucous membrane there. What's less obvious, I think, is the inside of your intestines are also kind of part of your outside, if you will. And I want you to think about it this way. If you think of the human body like a donut with a hole going through it, when you eat something and it goes into your stomach and then it goes through your intestines, it's inside you the same way that you can put your finger in the middle of a donut in the hole and you're inside the donut, but you're not inside the donut. Yeah? Okay, so you're in the inside, but it's still kind of, it hasn't gone through all the clear, like the security clearances that other things would need to go through if they went through your skin. Well, so this is how I think about the intestinal Peyer's patches. So, you know, at the, another analogy, at the borders between the US and Canada and US and Mexico, there's border patrol, right? There's border patrol agents and there's customs officials. There are also these things in the airport in Iowa, because flights can come in from international locations, and so they still need to go through security. Similarly, stuff that goes through your intestines also better clear security before it's allowed in. Amazing. All right, so lymphocytes are the cells of the adaptive immunity. They can move around all these different locations. So the B cells and the T cells, they can encounter the antigens either out in the tissues or in the bloodstream. They travel through the lymph nodes, in the spleen. The macrophages and the dendritic cells also go to lymph nodes. These are like, meetup locations, and they present their antigen. And when there is an infection, there's the proliferation, so we present the antigen, and then the T cells start making lots and lots of copies of themselves, and there's more of them. This is why the lymph nodes swell. This is why we check to see if somebody's lymph nodes are palpable. It's a sign of infection because we have all of these lymphocytes there trying to fight this infection. And in these sites, we have the helper T cells that have been stimulated by the antigen presenting cells. And then it's the helper T cells that go about activating the B cells and the cytotoxic T cells. All right, so adaptive immunity characteristics, highly selective, very specific. This is what I was telling you about the antibodies are actually shaped in a way that the tips of them are very specifically going to respond to a shape. So not every B cell is going to respond to every infection. Specialized to recognize a single antigen. Once the lymphocytes have been stimulated, they make lots of copies of themselves, and some of those copies are the so-called memory cells that stick around in case they're needed later, and when you're exposed to the same antigen. You've heard this before. We say there's two types of responses to the antigens. There's the humoral immunity, that's from the B cells producing the antibodies. And there is the cell-mediated immunity. This is by the T cells. And I'll tell more about CD8 cells in a moment. All right, so let's talk about B cells in more detail. And as I'm looking at this, this is getting kind of long, so this might be a good place to take a break, and we'll do a part 3 this time.